Hello everyone, my name is Shiraz Mumtaz. Today's topic is understanding the structure of skeletal muscle. Okay, um, so in detail we are going to study about what are the filaments, so what are the structures, and how we are going to see the thick and thin band. Okay, and some of the micro proteins as well. And then we are also going to see the understanding of the pathophysiology of the malignant hyperthermia. If one of the structures stop working or secreting something else, which is calcium, and how we are going to treat the malignant hyperthermia. Okay. So now, first of all, we have what you call a structure called the sarcolemma. Sarcolemma is the plasma membrane of the muscle fiber, which we're going to see in the next slide, which surround the sarcoplasm, which is actually the cytoplasm. And then the sudden change in membrane potential initiate the contraction. So right here we have the sarcolemma, and then inside we have the sarcoplasm, and then within the sarcoplasma we have different structure, the thick filament, which you can see in the blue color, and then thin filament we can see in the pink. And then while we have, like, these are this is the sarcoplasmic reticulum, this we have the transverse tubule or the T tubule, and then on the each side of the transverse tubule we have the terminal cisternae. The terminal cisternae actually is a part of the sarcoplasmic reticulum. Okay. So as we discussed it earlier, that we have the transverse tubule that go from the extend from the sarcolemma all the way toward the sarcoplasm. Actually, that is the one taking the current from the outside toward the inside of the cell of the muscle cell. So right here, as you can see, the sarcoplasm, the sarcoplasmic reticulum, which stores the calcium. As soon as the current, the action potential coming from the sarcolemma going into the inside, and then it is going to change inside the protein, which is in the sarcoplasmic reticulum, and then it is ultimately going to read the calcium within, and then we are going to see what calcium is going to do with the actin filament later. Okay, so as I said earlier, the tubular network of the each myofibril contain the terminal cisternae and the sarcoplasmic, which is the part of the sarcoplasmic reticulum. Okay, and then the sarcoplasmic reticulum that store and release the calcium whenever it's needed for the contraction from the terminal cisternae. Okay, so myofibrils we have what is it called? They contain the thick and thin filaments. Okay, and then the thick filament, thin filament called the actin filaments, and then the thick filament called the myosin filament. Okay, and then these are the, thick and thin filament are responsible for the muscle contraction. Okay. So now we have the structure called the sarcomere. Sarcomere, as we're going to see in the next slide, there is the smallest functional unit of the muscle fiber, the myofibrils. These filaments, that we are the thick and thin filament, are arranged into different bands, the dark band called the A band, and then the light band called the I band. Actually, we are going to see, mainly the dark band we are going to see the thick band, and then the light band we are going to see the actin filament, the myosin with the A, and then the actin. But you're going to see, in the dark band we are also going to see a thick and thin filament, which is going to see in the next slide. So right here, now we are going to see the structure called the A band that I said earlier, and that it has a thick band, okay, but it has a thick and thin band as well. The I band only contains the thin, which is the called the actin filament, the thick band, which is called only the myosin filament. In the meantime, we can see the M line, okay, which is what is it called? Giving the stability to the thick band, and then we have the H band, which is actually giving the stability on the H side of the what is it called, of the M line. Okay, and okay. So as I discussed it earlier, the A band contains the M line and the H band. Okay, the M line, which is the stabilizing the thick filament. Okay, and then the H band. Okay, which is on the either side of the M line. Okay. and then the H band only contains what is it called, the thick filament only, not the thin filament. And then the M line, which is, as I said earlier, which is only the stabilizing the thick filaments. Okay. We have the zone of the overlap, which is the dark regions, okay, which is the thick and thin filament overlap with each other, as I showed it earlier. And then we have the I band, which I said earlier. There is no thick filament in the I band. And there is a Z line, which is like this said earlier, that it is marked between boundaries from one from one sarcomere length to the other sarcomere. Okay. Tintin, that we have another protein which we're gonna show you next. This is the part of the elastic protein. It is how the muscle restored its original length, the sarcomere. So right here, which is the tintin, which is what is it called, the green line. Okay, so right here there's connecting between the Z line all the way towards that thick filament right here, and that has a property of the elasticity. Okay. The thin filament we have, okay, I'm going to show in the next picture, contain different molecule, F-actin molecule, the nebulin, tropomyosin, and troponin. Okay, and then the nebulin is holding the F-actin strands together. Okay, and then you're gonna see in different way, how the troponin and tropomyosin are holding those active sites on that G-actin protein. Okay. So right here, as I discussed it earlier, this is the nebulin protein that is holding the F-actin molecule. And then right here, as you can see, this is the troponin, and which is holding the active site on the G-actin molecule. And then ultimately, those strand of the tropomyosin is holding the the active site on that G-actin molecule as well. So whenever the contraction is happening, the calcium is going to come in. Okay, as I said earlier, slide, that the troponin, the calcium is going to come in. Calcium is going to react or act on the troponin, and the troponin is going to leave the site, and then ultimately this lock is going to open. Okay, troponin has a key. Okay, and then this is kind of a a chain that is connecting to the F-actin site. So whenever the troponin is going to leave the site, it is going to leave, the troponin is going to leave the active site on the G-actin molecule. Okay, and then it is going to leave its site for the myosin filament connect and then do the process of contraction. Okay, so right here we have the what they call again the continuing with the thin filament, as we discussed it earlier. The troponin is holding the active site of the G-actin molecule, and then also the tropomyosin connecting the active site on that G-actin molecule as well. Okay. All right. So continuing finishing with the thin filament, now we have the thick filament. A thick filament, as we're going to see in the next slide, it has the the 300 myosin molecule in then each the filaments. Okay, and then each molecule contain that tail and then the head, and then head is going to move actually towards the nearest thin filament, which is the active site on the G-actin molecule. Okay, so right here, as you can see, this is the tail, this is the head. The head is going to attach each side of the on the G-actin, on the active side of the G-actin molecule, and then doing its own contraction. Okay, so now we're going to understand about the fact of the malignant hyperthermia. Malignant hyperthermia, it is actually a genetic condition, which is going to be a mutation on the ROIR protein, which is going to come in next, within the sarcoplasmic reticulum. So the malignant hypertension causes the extreme response to the anesthetic drug, which is the succinylcholine most commonly. Okay, and the secondary symptoms is going to be the increase in the body temperature, and then the muscle stiffness, rigidity, and causing the tachycardia, which is increasing in the heart rate. Okay. In the severe cases of the malignant hyperthermia, this causes the muscle breakdown, okay, which is ultimately called the rhabdomyolysis, okay, which is the breakdown of the muscle, okay, and and causes the increase in the potassium, because since there is a muscle breakdown, this is going to increase in the potassium that is coming out from the muscle cells, and also because of it, because of the acidic environment within the muscle, within the cell, causes the acidosis. So, and then the increase in the potassium can also cause the problem in the rhythm, and if there is a rhythm problem, which can cause the cardiac arrhythmia, and then if these problems are not treated properly, it can lead to death. Okay, so as I said earlier, the malignant hyperthermia treatment is going to be now what we call treated by uh, the dantrolene, which acts on the receptor, the protein, the ryanodine receptor, RYR, and that is causing the block within the calcium channel within the sarcoplasmic reticulum. And then when there is no the release of the calcium, there is decrease of the what is it called, the contraction. There is no interaction with the troponin, as we discussed earlier, and then there is no interaction between the thin actin filament with the myosin molecule. Okay, and then that is how it is. This is the viral treatment for the malignant hyperthermia. So as I said earlier, this is the RYR protein. So dantrolene is going to get the negative effect. So calcium is going to, what is it called, decrease in excretion from the sarcoplasmic reticulum, and this is causing ultimately decrease in the what is it called, contraction of the muscle, and can save the person's life. Okay. Right here we have the analysis, like, of two cases, like, that when, like, dantrolene is used. Okay, in the first patient, which is showing in orange, that we, like, use, like, when did they use, like, it's around the fourth hour, like, around the fourth hour we use, like, or after the fourth hour they used. So it took them a longer time to patient to recover. As you can see right here, we have the potassium is what I call going up. Okay, and then the potassium, like, at that time is trying to coming down as soon as we hit with the dantrolene. Okay, and then creatine kinase, which is the breakdown of the muscle, is going up. Okay, so it happened, like, in the first case, like, we, like, they waited for a longer time. So in the second case, as soon as they discovered, like, within the 30 minutes, like, they discovered, so they administered their dantrolene. How it is affecting the what is it called, the patient. Okay, so it is not causing much of the muscle damage, and not of the release of the creatine kinase, and then the minimal release of the potassium, which means there's minimal damage going on. So it has proved that, like, that if we, like, found it earlier about the symptoms of the what is it called, the malignant hyperthermia, we can save the per patient's life. Okay, and then if later, like, around the fourth hour, or later than fourth hour, 4.5 hour we discussed. Okay, so it is going to be, like, creating more complications, and if not treated, like, hopefully not, like, then we can, like, we can lose the patient's life. Okay, so right here, so prompt administration causes the first, like, sign, the malignant hypertension reaction is critical and is more effective. Okay, so anyways, thank you very much.